So today we are going to discuss about reactions of monosaccharide. It's a very important topic in carbohydrate chemistry. So the first reaction in monosaccharide is oxidation reaction. So we know that carbohydrates are aldehyde. Carbohydrates contain either aldehyde or ketones functional group. That means the first carbon atom in a carbohydrate molecule will be an aldehyde and the last carbon atom will be an alcohol group. And if it is having the functional group ketose, the second carbon atom will be possessing the functional group ketose and the last carbon atom will be having the functional group alcohol as the functional group. So depending upon the oxidizing agent used, the aldehyde or the alcohol group can be undergo oxidation. So if you are using mild oxidizing agent, under that condition, the aldehyde group, you can see in this picture here, glucose is given. So if you are using mild oxidizing agent, the aldehyde group which is present in the glucose is oxidized to carboxylic acid, that is resulting in the formation of aldonic acid. You can see here glucose is converted. Here the mild oxidizing agent used is bromine. In presence of bromine, the glucose is get converted to gluconic acid. Okay, like that. Under certain special condition, the aldehyde group will be protected and the molecules will be undergoing oxidation and the terminal alcohol group will be oxidized to carboxylic group. That means when the aldehyde group is protected and the molecule is undergoing oxidation, the terminal alcohol group will be converted to carboxylic acid and will result in the formation of uronic acid. Okay, so the glucose will be oxidized to glucuronic acid. We know the importance of glucuronic acid. It's a very important acid which we use in conjugation reaction. We are using this in conjugation reactions. Then mannose is converted to mannuronic acid. Like that, galactose is converted to galacturonic acid. So this is occurring under special condition. Under certain special condition, the aldehyde group will be protected and the terminal alcohol group will be oxidized to corresponding acids. Okay, that is resulting in the formation of uronic acid. Okay, then when we are using strong oxidizing agent, what is happening? The first, that means strong oxidizing agent means nitric acid. Like that, when strong oxidizing agents are used, the first and the last carbon atom will be oxidized. Both the first carbon atom and the last carbon, that means the functional group, the aldehyde functional group and the alcoholic functional group will be undergoing oxidation to corresponding dicarboxylic acid, and the dicarboxylic acid which is formed as a result of oxidation under strong condition is called as saccaric acid. So glucose will be oxidized to glucosaccaric acid. Mannose will be oxidized to mannaric acid. Like that, galactose will be oxidized to mucic acid. And this is a confirmatory test which we used to for the mucic acid. Galactose is converted to mucic acid. This is a confirmatory test which we used to for the conformation of galactose, the presence of galactose in a particular compound. We use mucic acid. In that, galactose will be converted to mucic acid under strong oxidizing condition. Both the aldehyde group and the alcohol group will be undergoing oxidation and will be get converted to the corresponding dicarboxylic acid. So that is about under strong oxidation condition. So this is about this oxidation. Mild condition, what is happening under special condition, and what is happening under strong oxidizing condition. Now we are moving on to the next reaction, that is reduction. When we are treating this with a reducing agent such as sodium amalgam or like that, the aldehyde or the ketonic group, of the monosaccharide is reduced to corresponding alcohol. Here you can see that is giving the CH double bond R, that is a functional group, aldehydic functional group is given. And when it is treated under with a reducing agent such as sodium amalgam, it is undergoing reduction. So what is happening here is the monosaccharide, the double bond, oxygen, C double bond O, in that double bond will be getting broke, and the O is get converted to OH and a hydrogen atom is added. So it is get converted to CH2OH. Okay, so Under reduction, the aldehyde or keto group of the monosaccharide is reduced to corresponding alcohol, and it is indicated in that general formula. Then like that, the important monosaccharide and their corresponding alcohol, when it is undergoing reduction, the corresponding products are given formed. You can see in the picture that glucose will be get converted to sorbitol. Then like that, galactose will be get converted to dulcitol. The mannose will be get converted to mannitol. Like that, fructose, in case of fructose only, two compounds is formed, mannitol and sorbitol is formed. Okay, then like that, ribose will be get converted to ribitol. Okay, so that is about the important monosaccharides and their corresponding glucose will be get converted to sorbitol, galactose will be get converted to dulcitol, mannose will be get converted to mannitol, ribose will be get converted to ribitol, like that. Okay, then this is about reduction reaction. Then what is the clinical significance? Is there any clinical significance about the alcohol which is produced as a result of reduction reaction? As there is very in a clinical significance is there, that is the sorbitol and the dulcitol, which is sorbitol is produced by the oxidation of glucose, and dulcitol is produced by the oxidation of dulcitol is produced by the reduction of galactose. When this sorbitol and the dulcitol is accumulating in the tissues in large amount, that can cause strong osmotic effect. We know that when it is causing strong osmotic effect, that is leading to the swelling of the cells. Okay, the cell, there will be chance of the cell lysis can take place. Okay, there will be swelling of the cells, water will be getting accumulated inside the cells. So under certain pathological condition, for example in the case of cataract, peripheral neuropathy, nephropathy, like that condition, the sorbitol and dulcitol can get accumulated in the tissues in large amount, can cause the strong osmotic effect, and that will be leading to the swelling of these cells. Okay, then mannitol, which is the reduction product of the sugar mannose, that will be useful to reduce intracranial tension by forced diuresis. So this is about some of the clinical significance which is produced, clinical significance caused by the product when it is undergoing products, that is the carb sugars, when it is undergoing the products of sugars when it is undergoing reduction. Okay, then next we are discussing about another important reaction of monosaccharide, that is osazone formation. In the case of orsazole formation, we know that phenyl hydrazine is a reagent we used. Then when phenyl hydrazine in acetic acid is, that is when the phenyl hydrazine reagent in acetic acid, and if we boiling this, or if we are taking some reducing sugars, and when we are adding this phenyl hydrazine reagent and acetic acid, osazones are formed. What do you mean by osazone? Osazone means at first when the phenyl hydrazine reagent is reacting with the glucose molecule, hydrozones will be formed, that is soluble form. That is soluble, but this glucohydrazone will again react with the phenyl hydrazine molecule and will be get converted to osazone, that is insoluble in nature. So that insoluble condition is actually forming crystals like structure. And the crystals can be of various shape. Okay, that means in the case of maltose, if the reducing sugar is maltose, sunflower shaped crystals will be formed. If it is in the case of glucose, needle shaped crystals will be formed. In the case of disaccharide like lactose, powder puff shaped crystals will be formed. Okay, so these are some of the shape of the crystals which is formed as a result of what? Osazone reaction. So these are about the reactions of monosaccharide. So we have discussed about oxidation reaction, reduction reaction, and we have discussed about ozone formation also. Okay, thank you.